All right. Good afternoon, physiology. I hope you guys have had a good week since the last time we met. We are down to the last two virtual meetings, fingers crossed. So I don't know about you, but I'm looking forward to actually getting back in the lab so we can actually do more complicated labs. I'm glad to see that you guys tried the coloring. I hope you enjoyed that while you were waiting for me to get everything ready. Unfortunately, I have to erase all of this stuff. You couldn't hear me at first. Can you hear me now?
Yes.
Okay. I don't think I was saying anything all that important. Just
okay.
Just hi, and 2 weeks from now, we're in the lab.
Okay.
Fingers crossed. Okay, so this is more important here. Lab 4, part 1, and the quiz are due Sunday, February 6th before midnight. Next week, we're doing a sensory lab. We're going to do part of it as a group and part of it on your own. So I will post some documents that you will need to print out. And I will post a list of things that you will need to have handy to do a couple of those labs. It's all stuff you should have around the house, like ice water. So you shouldn't have to go out and buy anything for it. I hope not anyway. But I don't have those up yet. Those will be up over the weekend. Okay, so the lab that we're going to do today is on DNA and protein synthesis. So the idea is to help you understand DNA, messenger RNA, tRNA, rRNA, and how they work together to make proteins in the cell. So we're going to kind of walk through transcription and translation, the two halves of protein synthesis. And we're also going to look at the effect of mutations on protein synthesis. So this is a basic DNA model. If you look on the left, this is a single strand of DNA. You're probably more familiar with the image on the right, where two strands come together and form the double helix. The backbone is made of alternating sugars and phosphates. I'm sure a lot of this is very familiar to you guys. And the two strands are held together by base pairs. And the base pairing is very, very specific. Adenine binds to thymine, and cytosine binds to guanine. So the mnemonic here is apples grow on trees, adenine and thymine, and cars park in garages, cytosine and guanine. All right, I just told you this. Did everybody pay attention, I hope? Oh, didn't quite pay attention. The phosphate and sugars, they're the backbone of the DNA strand. So they're the sides of the ladder. So if we look on the double helix and we think of this as a ladder, these are the sides and they're sugars and phosphates alternating. And cytosine bonds with guanine. Looks like almost everybody got that one right. Okay, so the terminology might have been confusing because you may not see that double helix as a twisted ladder. It's described that way a lot. I've also seen it described as a spiral staircase. Same kind of concepts. The sugars and phosphates make up the sides of the ladder, and the bases make up the bonds. Now, the order of bases makes up the information of DNA. So the sequence of those bases, that describes what protein is going to be made. And that's the information needed for your cells and your bodies to function. We also say, could also say the sequence of bases code for the sequence of amino acids in the protein. And the sequence of amino acids in the protein determines the structure, the shape, and the function of the protein. So hopefully you can see that that sequence of nucleotide bases in the DNA really determines the final shape and function of the protein. I hope a lot of this is a refresher for you guys. A lot of people learn this in basic bio and then forget it. Everybody keeping up? Okay, good. All right, so the first step in protein synthesis and making a protein is DNA is transcribed into RNA, specifically mRNA. Does anybody remember what the M in mRNA stands for?
Messenger.
Messenger, perfect, thank you. So DNA, I'm sure you remember, in eukaryotic cells stays in the nucleus. We say it's transcribed into RNA. When you transcribe something, you are copying it over. So, for example, you may transcribe your notes. You may copy notes from somebody else. That's kind of where that word comes from. So the information stays the same, but RNA is slightly different than DNA. So RNA is less stable. It doesn't last as long, but it can move out of the nucleus, which DNA can't do. Generally speaking, RNA is a single strand. That single strand can be in a lot of different shapes. So, oops. So it can be kind of just a string, which is what we think of for mRNA. But as we'll see, it can also have some seriously funky shapes. Kind of like that. That, as we will see, is a tRNA. And while we're going to be talking about tRNAs and ribosomal RNAs and messenger RNAs, there are other RNAs out there. And a lot of those other RNAs are going to have very different functions in the cell, not just protein synthesis, but for example, a major role for RNAs is cellular defense against viruses and bacteria. So the cell can make RNAs that will bind to the genetic information on a virus or a bacteria and prevent it from functioning or signal it for being broken down. So there's all kinds of things that go on here beyond what you're going to be learning here. Okay. Besides being a single strand, RNA has a different sugar. So DNA has deoxyribose. RNA has ribose. And the difference is right here. So if you're not comfortable with chemical drawings, this is ribose sugar here. There's a carbon at this intersection, an oxygen here, a carbon here, a carbon here, and a carbon here. This carbon is connected to a carbon and a hydrogen. This carbon to an oxygen and a hydrogen. This carbon to a hydrogen and an oxygen. This carbon, it's not written in, but there's a hydrogen here, as well as bonding to the neighboring oxygen and the neighboring carbon. The difference in deoxyribose, that oxygen at this particular carbon is missing. DE means without. Oxy refers to oxygen. So it's a ribose sugar that is missing an oxygen. And the other big difference is in the nucleotides. So while DNA has thymine and cytosine and guanine and adenine, RNA has cytosine and guanine and adenine, but has uracil instead of thymine. So it's a slightly different molecule. It does bond with adenine. It kind of does a function basically like thymine. So our double strand of DNA in the nucleus gets copied to a messenger RNA that moves out of the nucleus. And once it moves out of the nucleus, it connects. I'm not good at drawing on the whiteboard here. It connects to a ribosome. And we're going to go on to that in just a second. This first part is transcription.
I have a quick question.
Yes.
So since the mRNA is just like one strand, does that mean it copies like half of the DNA and there's another one for the other half?
Oh, that's actually a really good question. It's more complicated than that.
Mm-hmm.
So let me see if I can clear this and I'll draw it out for you because it's easier to draw it than to say it. So here is your double strand of DNA. In order for transcription to occur, you have to form a space, a bubble where the bonds between the bases are broken. Only one side will be copied to make mRNA. We're not copying both sides, we're just copying one side. If we copied both sides, we would end up with an mRNA that's backwards. For slightly more complicated reasons, but this is the basic idea. You're only copying one strand. All right, so the next step of the process is called translation. Actually, let me go back to the whiteboard. Translation converts the language of DNA and RNA into the language of amino acids, proteins. So we're translating between languages. It's taking the information in the messenger RNA and using that to create a sequence of amino acids, which eventually become a protein. So DNA transcribed to RNA, which is translated to protein. This DNA to RNA to protein is called the central dogma of molecular biology. Not that I'm going to ask you that, but if you hear about the central dogma, that's what we're talking about. Now, we only have 4 bases in DNA and RNA. In order to code for the 20 amino acids that are commonly found in our bodies, we can't just have a 1-letter to 1-letter correspondence. It doesn't work that way. So what evolution has done is created the codon. So a codon is 3 nucleotides in a row that code for a single amino acid. So for every amino acid in a protein, you're going to have 3 bases or 3 nucleotides in the genetic information, in the DNA and in the messenger RNA. This is a codon table. The way this is organized, on the left-hand side, you find the first letter of your codon. So if you're reading a DNA sequence and it goes AUG, CAC, UUG, this is messenger RNA, right? Because it's got the uracil. We start with our first letter. Here is A. Across the top is the code for the second letter. So our first column has the U. And then in the box for A and U, we see all 4 options for that third letter. So AUG codes for methionine. I am not going to expect you to memorize or learn the amino acids. You may have to do that at some point in your careers, but not for me. All of the amino acids have 3-letter codes to abbreviate for them. So methionine is MET. So if I'm asking you to use a codon table, I would expect you to write for AUG MET. What would CAC code? What would the amino acid be? Just using the codes on here. His, that's right. Histidine. And then UUG. There's the first U. There's the second U. There's the box where all of the codons start with UU. What's Leu? Leucine. Yep. So these 9 bases code for these 3 amino acids. That's a huge arrow. All sequences, messenger RNA sequences, start, how can I put this? The protein synthesis always starts with AUG. So all proteins start with a methionine. Now, sometimes it's processed and that's cut off later, but that's how manufacturing always starts. You guys might want to take a picture of the codon table here or a screenshot. There's another way of showing this, which is the wheel. It's still kind of called a codon table, but in this one, you start in the center, and we're starting with AUG again. So A, all of the codons that start with A are in this quadrant right here. There's A. The next one is U. So all of the codons that start with AU are in this area. And there's G, which is methionine and start. Everybody learns a different version of the tables. What would GAG code for? Glute. Yep. GLU. And CUA? Lou, good. Glad to see somebody new chiming in. And UAA. Stop. Yes. There are certain codons that indicate that that is where translation stops. So that lets the ribosome know that it's time to stop making that protein and to release it. So you'll notice the 5' and the 3'. What in the hell does that mean? Okay, this is a little hard to interpret. DNA and RNA have a 3' and a 5' end. So here's a short 4 nucleotide string of DNA. It's got a 3' end and a 5' end. What that means is when we look at the sugar in the nucleotide, the carbons are numbered around the ring structure. The carbon that is connected to the base is carbon number 1. And then we go clockwise. We get carbon number 2, 3, 4, and 5. 5 is not in the ring. It sticks up above the plane of the ring. It sticks out. So if carbon number 3 is not attached to a phosphate group, that is the 3' end. And if carbon 5 is not connected to another nucleotide, we call that the 5' end. And why do we care? We care because we have to know which direction to read the gene or the messenger RNA. And so we read messenger RNA starting with the 5' end and going to the 3' end. And those codon tables use the codes on the messenger RNA, not the DNA. So DNA has 3' and 5' end, but remember you've got 2 strands? Well, we'll just read the messenger RNA rather than try and sort through the DNA sequence and which end of the gene starts on and that kind of stuff. Okay. So, translation. Transcription is pretty straightforward. Because cytosine always binds with guanine, you take your DNA and everywhere there's a thymine, a guanine comes in. Everywhere there's an adenine, a uracil comes in, and you just kind of construct it step by step. Translation is a little more complicated. It has 3 major steps: initiation, elongation, and termination. So in this image, this bar is our messenger RNA. And this kind of beige colored part is the ribosome. So a ribosome has 2 subunits called small and large. Not very creative there, but it's not anything that's hard to remember. And when the first transfer RNA, we haven't talked about transfer RNAs yet. tRNA is transfer, and those bring the amino acids to the ribosomes. So when the first tRNA attaches to a messenger RNA, the large subunit then attaches to form the fully functional ribosome. So the ribosome is in 2 parts, and only when that first tRNA is attached does the large subunit come in to form the fully functional ribosome. And the first tRNA, as we've said, is always methionine. So that's attached. The ribosome, you can picture it moving one codon down the strand of the mRNA. So elongation is a series of steps where the ribosome moves one codon down the mRNA toward the 3' end. It moves one step down, and then your next codon is available. So only one codon is available for a tRNA to fit in there at any given time. So the ribosome moves down, the next codon on the mRNA becomes available, and here comes a tRNA with a phenylalanine. And that will attach. You'll get a bond forming between the amino acids and the ribosome moves another step down, one more codon down. When it does that, it releases the first tRNA, which is now empty. It doesn't have an amino acid on it. Initiation. So let's talk about exactly what goes on in each of these. Initiation. First tRNA attaches to the mRNA. Well, let's just say the tRNA attaches to the mRNA. Let's leave off the first because this happens for the second one and the third one too. Elongation. The ribosome moves down the mRNA. Another codon is exposed. Another tRNA comes in. The amino acids bond together. Termination. The ribosome moves down, and the first tRNA, or the first one in this series of steps, is released. All right, this is a little complicated. Who has a question for me? Who wants me to go back over something? Will this lab be posted on Canvas? Yes, it will. It doesn't have the amount of detail as what I'm talking about, though.
So the bottom, the last part, right there, yeah.
Mhm.
So that's the initiation, right? Or no?
Okay,
Sorry.
for this tRNA, the one that's empty, this is termination. But this amino acid with the arginine is initiating.
Okay, I see. And then the middle one, is that elongation?
Yes, yes.
Okay, that makes more sense.
Yep. Like I said, it's kind of complicated because all of these steps are going on at the same time. And this goes until you get to a stop codon. Now, how does the tRNA know where to attach to an mRNA? So remember that kind of funny looking clover leaf. That is a tRNA or transfer RNA. And at the bottom of this, it has 3 nucleotides called an anticodon. And if we've got our mRNA here, we have 3 nucleotides that make up the codon. And the anticodon is exactly what you think when you're hearing it. It's a series of 3 nucleotides that can form those bonds with the 3 nucleotides in the codon. So if the codon is AUG, the anticodon is going to be UAC, because adenine bonds to uracil, uracil bonds to adenine, and guanine bonds to cytosine. Is everybody following this? Is anybody following this? Dead silence. Okay. I got a yes. Thank you. All right. This is the kind of thing that you're going to see in the lab assignment. You've got a sequence on a DNA strand. And I'm going to ask you to give the sequence of the messenger RNA strand, the corresponding sequence on the tRNA, and then I'm going to have you guys use an amino acid table to identify the amino acid. So let's see if I can mess with this. See if I can put it on the same page. Oh, no, of course not. The formatting's wrong. I was going to try and put it on the same page as our codon tables, but the formatting screws up. Yes, this is for you to fill out, and well, you don't have to print it out now because there's a copy of what you need on Canvas, but that is what you are going to be filling out on Canvas. So let's go back to the PowerPoint and practice this. So I'm going to write out a DNA sequence. Oops. So translate that into an mRNA sequence. Anybody want to tell me what the mRNA sequence is going to be, or put it in the chat? No, nobody wants to volunteer. Okay, in that case, when we look at this lab, write down this sequence.
I'm a little confused on something.
Okay.
Can I ask a question?
Yes, yes, absolutely.
Okay, so I was trying to figure it out and I got like the gag one, but not the TAC. Because there's no T on the chart. Because I took a picture and I was looking at it. So I didn't know what to do for that.
Right. Okay, I probably didn't explain this very clearly. So what you need to do is take the information here and translate it into an mRNA sequence. So T, which is on DNA, binds to A. So the mRNA strand is going to have an A there. A bonds to U, so the mRNA strand will have a U, and C bonds to G, so the mRNA strand will have a G. So that's AUG.
Oh, okay. And to figure out the A, is that the, because I wrote down like the apples grow on trees, cars park in garages.
Yes.
Okay. And then how did you get the U from the A?
Because we're going from DNA to RNA, and every time a DNA strand would have a T, an mRNA strand will have a U. Because all RNAs use uracil instead of thymine.
Okay, that makes more sense. Thank you.
So the anticodon on the tRNA, A bonds with T, but RNAs have U instead, so that would be U. This U would bond with an A, and this G would bond with a C. This is where we use our amino acid table. Unfortunately, I can't show you both of these at a time, but we read the mRNA strand and we translate that to an amino acid sequence. So now you take, Jess, go ahead.
So when you're bonding, or when you're translating the mRNA strand from the DNA strand, you have the whole T to A to U, and then for the anticodon, you would still use the same rules and translate it that way.
Yes, except we are going to say transcribe.
Oh, transcribe, sorry.
No problem. It's new
All right, just a
terminology.
clarification point, yeah.
Yeah. So transcribing, we're using those T and A and C and G rules. Translation, we're going to the amino acid. And that's where we use our codon table. So we use the codon table to fill out the amino acid line. And we read the mRNA. So the mRNA strand has a codon AUG, and that codes for methionine. Is that a little clearer? Don't worry about the last column yet.
Makes
Uh,
sense.
That makes more sense.
Okay, good, good. Thanks for chiming in and asking the questions, because if you're confused, I'm sure a lot of you guys are confused.
So the mRNA is what codes for the amino acid.
Yes.
Okay,
Yes.
got it. Thank you.
All right.
I'm still a little confused on how the tRNA fits in. Does the mRNA go to the ribosome or the tRNA?
Both.
Do they go to two different ribosomes? Or it does, I guess it doesn't matter.
They go to the same ribosome. Let me go back
Okay.
to see if I can remember where that image was. Okay. You know what? I'm gonna what I'm gonna do, I'm gonna explain that. I am going to explain that.
Mhm.
What I'd like for you guys to do is look at this image and figure out what the mRNA strand is going to say. And what I'm going to do is I'm going to go look for a translation video real quick, because I think it'll really help everybody understand that. So do you guys want to work on this on your own, or do you want to be in breakout groups?
I think it'd be fine just on our own.
On your own? Okay. I'm going to shut off my mic for a couple of minutes while you guys are doing that. How you guys doing on that?
Not bad.
Good? Okay, great. So I'm going to move us to a video. And for some reason, when I try and Zoom videos, a lot of times the sound does not work right. But I've got closed captioning on. So this is a video. They've already talked about a whole bunch of stuff, but I wanted to show you this part with the ribosomes. Notice there are 3 spaces here in the ribosome. That is important for translation. So this ribosome already has the small and the large subunits. And it does have a type of RNA called ribosomal RNA.
We can't hear anything.
Yeah. Okay. I figured that's what tends to happen. So, whoops, I went too far back. Where did I go?
Could you slow it down so we could read the translation?
Unfortunately, I don't know how to do that. So let's just kind of go step by step, and I'll just keep pausing it. So the ribosome, the rRNA, the ribosomal RNA, and the protein work together for translation. What the RNA does is it's involved in creating these 3 binding sites in the ribosome. So 1, 2, and 3. All right. This brings in the messenger RNA. And the messenger RNA lines up. It's not well done in this graphic, but it lines up with 1 codon in each of these 3 pockets. The first pocket, let's see if I can draw on this. So this is the first pocket. And what happens there is initiation. The tRNA comes in and attaches. So this is the tRNA. See this part here at the bottom? That is your anticodon. So that is going to bind to the codon in the initiation pocket. Let's see if he actually shows this. Oh, nope, he doesn't. He goes on to ramble. Okay, so then your tRNA comes in here. I'm just going to draw it as a kind of stick, and it's got the methionine. Then the ribosome is going to move 1 codon. Oh. Oh. Somebody apparently knows a trick to YouTube that I don't. Ho ho, look at that. I can slow it down. Let's see if this actually works. And clear my annotations off. This guy talks really fast. Does that make it a little bit clearer what's going on here?
Yes.
Yes, it does.
Good, good. Some things are just so much easier to animate than to explain. Like I mentioned in the chat, I will post a link to this video. It's got a lot of detail, more than what we're covering exactly, but it's useful. These crash course videos are usually pretty good. Okay, so in the lab that is online, you will need to transcribe the DNA information into mRNA. So let's look at starting here. if we translate G to messenger RNA, what's going to be on the messenger RNA next to a guanine?
The C.
Cytosine.
Yep, that's a C. And then for A,
U.
U. Ooh, boy, that's crappy drawing. Write it at the bottom where I'm not going to run out of space. Come on. And then the next G,
C.
C. Perfect. Now, as I mentioned, you will find this on Canvas and you will have plenty of time to do this on your own. We're just walking through part of it right now to make sure you guys understand how to fill this out at home. All right, so if this is the codon, what would be the anti-codon on a tRNA?
For the CUC
For CUC,
would be G, A, and G.
GAG, exactly. And to find the amino acid, we take CUC and we go back to the codon table. First letter C, second letter U, third letter C. Our amino acid is going to be leucine. And so on the blank here, leucine. Now, the idea is that you're going to fill out that table and then you're going to make a string of paper clips. Now, does everybody have colored paper clips? No, of course not. So you can write in the color. You can either string paper clips together, or you can take some kind of markers and mark out basically a string of all the different colors. The colors are useful because after we do the first sequence here, then we're going to have a mutation. So we're going to have two kinds of genetic mutation, and we're going to see the effect on the amino acids. And if you've got colored paper clips, or if you've got crayons and you can color little circles on a piece of paper, there's a visual way to see these differences. The first one, and I'm going to talk about these different kinds of mutations, is a simple base substitution. So here we have a guanine, and at the same spot in the original DNA was a cytosine. So that cytosine changes to guanine. Is that having an effect on your amino acid sequence? Is that having an effect on your paperclip color? And then the other one is inserting a base in the DNA sequence, and what effect that's going to have on your amino acid sequence and your string of paper clips. So when we go back to our codon table, notice that if we look here, oh my goodness, look at this part of the graph. Any codon that begins with CU is going to code for a leucine. And in fact, if it starts with UU and ends with either A or G, it also codes for a leucine. There's a lot of variability, multiple codes for a given amino acid, which means if you have a codon that reads CUU and a mutation occurs in the third position, and then you end up with CUC instead, you have the same amino acid in the protein. It's not going to have an effect. We call those silent mutations. Now we do have a couple of exceptions. So UGG codes for tryptophan. It is the only sequence. So if there's a mutation in this codon, it will change the amino acid. AUG only codes for methionine. If you have a mutation in that, you're going to have a different amino acid. And of course, if you have a mutation in the first two columns here for leucine, that could cause you to have a proline or a histine or a glycine or an arginine. So there could be lots of different amino acids that could end up in that spot in the final protein. So what this means not all genetic mutations change the amino acid sequence. Which means that not all genetic mutations are going to have any kind of effect on the cell or the organism. And what I didn't put in here is, shoot. Let's go back to this window. Here's what we want. I'm going to copy this back into the PowerPoint so that we can manipulate it and mess with it. And then okay. So these are looks like that might be all of the 20 amino acids. We can group amino acids based on characteristics of those amino acids. So up on the top, we're talking about amino acids with charges on the side chain, which means that they are either acidic or basic. So these act like a alkaline chemical or a base. And these act like an acid. So they've got a charge on them, and that's going to affect what they do in the protein. This group of amino acids, their side chain is not charged, but it's polar. Polar means hydrophilic. Which means they're going to be wanting to be exposed to water. The cytosol has a lot of water. Cell membranes don't. So these are going to be wanting to be exposed to the cytosol. This bottom one, those are hydrophobic. Which means they're going to be on the interior of the protein or embedded in a membrane. So a protein that has a lot of hydrophobic amino acids in it is more likely to be a membrane-bound protein, one that is stuck in the membrane. Now, if we look at these hydrophobic amino acids and we look at isoleucine and leucine, there's not a whole lot of difference here. This part, of course, is identical, but this bit down here at the bottom, not a whole lot of difference there. If you had a mutation that changed an isoleucine to a leucine, do you think it's going to have an effect on the protein? Probably not. Now, if you changed an alanine to a tryptophan, tryptophan is a huge amino acid, and that's going to affect the shape of the protein because alanine is very, very small. And if the protein needs that alanine to be, surrounded by other amino acids, a tryptophan might not fit there. So when we think about genetic mutations, many of them have no effect, none whatsoever. If there's an effect, it's most likely going to be negative. And that's what we generally think of when we think of genetic mutations. We think of harmful ones. The rare ones are actually helpful or beneficial. And those are very, very important in evolution, for example. You guys following this part?
Yes.
Thank you. And just for your information, this table has the amino acid name and the 3-letter abbreviation. Amino acids also have a single letter abbreviation. I don't have those memorized. I have to look those up. I can usually tell you what they are from a 3-letter abbreviation though. So they exist. If you get into proteins, you'll probably learn them, but otherwise not so much. Okay. Let me clear those annotations. So DNA mutations can happen at any time. We all have cells in our body that have mutations. It's very, very common. When we looked at the handout, the worksheet that you guys are going to do on your own, we had a change in one codon to a different codon. But there can be other kinds of changes. So this is a hypothetical chromosome that has these 6 segments. These are larger genetic mutations. The larger the genetic mutation, the bigger the effect on the protein and therefore the organism. So this first image has lost the B section. So it's got a huge deletion. You can delete a single amino acid, you can delete a chunk of chromosome. If you delete a chunk, it's going to be worse. This next one over, the B got duplicated. So now you have 2 copies of this section. Well, that can be a problem too. It can be a huge problem. If you have twice as many genes, you may end up with twice as much of a specific protein. And a lot of proteins function on a dose-dependent manner, meaning how much of that protein is working determines how the cell's going to function. Down here, we have something called a translocation. So this is one chromosome, and down here is a second chromosome. And you can see that part of this second chromosome exchanged places with part of the first chromosome. So they swapped genes. And again, this can be problematic because you're taking these, the genes that have moved off of the original chromosome, which means they might be separated from the appropriate regulation. All of this crap is highly, highly, highly regulated. Everything in the cell is highly regulated. And if you screw up the regulation, that can be catastrophic for the cell. And then here's the last one. This is an inversion where the B and C on the left side ended up on the right side of the centromere here. And the D and E that were here ended up on the left side. And again, that is screwing up regulation. Okay. How does everybody feel about this right now? Do you think if you go to the lab, you'll be able to fill this out on your own? Oh, right, good. Okay. If you get confused and can't figure something out, feel free to email me. Feel free to watch this video again. Feel free to watch the crash course video. Just reach out to me if you're having problems and you need help. Next week, we're going to be doing senses. I'm hoping that that one's going to be a pretty fun one. And then we're going to be in person. So I will see everybody next week.
Thank you.
Thank you, Professor.
Thank you.
Bye, everybody.
Professor, when will you post the assignment that you were showing us?
I thought